Hi, my name is Jingfang Ju. I'm the program director of oncogenic drivers and mechanism of carcinogenesis at the Stony Brook Cancer Center. And I'm also a professor in the Department of Pathology. And in this Making Sense of Science session, I'd like to share with you a brief summary of our development of a microRNA-based platform technology recently for cancer therapeutics. My laboratory have a long-term interest to understand the mechanism of protein translation mediated by these microRNA in cancer, which I will allude to you later. And we like to understand the roles of microRNAs in chemoresistance, which is the major bottleneck for successful outcome of chemotherapy or radiation therapy, which we try to understand the role of microRNAs in regulating cell growth and cell death, such as programmed cell death we call apoptosis. Autophagy is another mode of cell death. EMT is a mechanism for cancer cells moving out of original position to metastasis sites. And we like to develop cancer biomarker based on microRNAs, and also we like to develop microRNA-based experimental therapeutics. Before I go into the details, I'd like to introduce the central dogma of human genetics, which I think will help understand this whole lecture. The central dogma of human life, or any life, is the genetic information flow from one direction, from DNA, which is the genetic information, to RNA through a process called transcription. And RNA is a template for protein synthesis, which is the process called protein translation. As you could appreciate, there's 3.2 billion base pairs in a single cell. And that translates to 2 meters long DNA. And you can imagine in our human body, there's 724 trillion cells. And that adds to 10 billion miles of DNA, which could circle from Earth to the sun 60 times. You could imagine this information is enormous. It could be equivalent to the entire universe. So in the sense, our human body is a universe. And the genetic makeup is basically by 4 building blocks, A, G, C, and T, which I will explain that why those are important. And you could see the task of understanding cancer is also very complex. And why we're interested in microRNA. What is microRNA? As the completion of Human Genome Project, a surprising discovery is that only 3% of our genome is template for protein synthesis. Over 97% of the human genome are so-called non-protein coding. They don't code for messenger RNAs. They are not template for protein synthesis. People used to call them junk genomic material. However, nature don't make junks. I think we recently discovered that these so-called junk RNA or so-called non-coding RNA play an important role in gene regulation. One of them is a short RNA, we call it microRNA, which I'll tell you why they're so important. So microRNA is a small RNA with the length about 20 nucleotides long. It's a short piece of RNA. You can see from this slide, it looks like a hairpin. It gets transcribed inside the nucleus as a precursor microRNA. And that gets exported to the cytoplasm of a cell, and that gets processed to a double-stranded RNA, which you could see on the left-hand side, a duplex, about 20 nucleotides long. That duplex, so-called mature microRNA, is able to interact with a messenger RNA template, which we call it target messenger RNA. By interaction, direct interaction, it suppresses protein synthesis, either by degrading the protein or by inhibiting the protein machinery to move down from the messenger RNA or cause the damage of messenger RNA. In a way, I draw a cartoon on the right-hand side. It's like a train track moving a train from one place to another which is the machinery for making protein, and their signals on the train track tell how fast or how slow this machinery is going to move down the track. And this is important. The microRNA play the important role as these signals that regulate the amount of protein that's needed for carrying the cellular function. As we all know, it's the protein that carries the function inside the cell, such as insulin that digests sugar, or growth factors, which are the proteins that regulate cell growth, or suppressor proteins that stop proteins. So in a way, it's like a car has an accelerator and a brake. The growth factors is like the accelerator, and the brakes are, we call the suppressor genes. And many of these microRNAs act as either an oncogenic RNA or a tumor suppressor RNA. So you could imagine why they are important, why we care about this. One of the mostly widely used chemotherapeutic drug we call 5-fluorouracil, 5-FU. And this 5-FU was initially synthesized and discovered by Dr. Charles Heidelberg in the 1950s. And this is the drug that's still used today for treating many solid tumors. And you can see from this biochemical pathway that this drug gets metabolized. The scientists spend years of studying this biochemical pathway. Without going through the details, I could show you that we understand the protein target, which is called TS. You could see in the middle, the 5-FU gets metabolized to a molecule called FDUMP that forms a suicide protein complex. It basically shuts down the TS protein function, which this TS protein convert U to T, which I mentioned there are 4 building blocks in our genome, A, G, C, T. This enzyme protein regulates synthesis of one of them called T. If a drug blocks these building blocks, you could imagine it will cause the depletion of one of the building blocks T that will cause DNA damage, which how this drug works to kill the cancer cells. However, it is extremely difficult to eliminate all tumor cells, and this slide shows that all tumor cells are not created equal. We call it heterogeneous, so the population of cancer cells are highly heterogeneous. And this cartoon basically shows the chemotherapy or radiation or targeted therapy often quite effective eliminating most of the rapid proliferating tumor cells. However, there's a small fraction of tumor cells, we call them cancer-resistant cells or tumor initiation cells or stem-like cells. These cells are highly resistant to chemotherapy or radiation. As a result of that, once patients stop treatment, the tumor eventually grows back, and those cells are highly resistant. And we learned that the microRNA play an important role in this process by regulating the phenotype, which is the phenomenon of resistance. By understanding this process, we try to come out a therapy based on the microRNA. One of the microRNA I provide you in this short summary is called miR-15A. It is a microRNA has a tumor suppressor function. The reason why we are interested in this microRNA is because tumor cells oftentimes have the reduced expression of this microRNA or completely lost the expression of this RNA. Basically, it's like the car brakes got damaged and the cells can grow out of control. So how are we going to repair this? We're going to restore the brake function by restoring this microRNA 15A into the tumor cells. And this is basically the data, a snapshot I want to share with you, how we understand this microRNA function as a brake. And we identified their oncogenic target proteins, which those proteins are basically the accelerators to push the cancer cells grow out of control. And by restoring the function of microRNA 15A, we could suppress the cell proliferation, as well as get rid of these resistant cells, As you could see at the bottom of this slide, there's spheroids, clusters of colon cancer stem cells that get completely eliminated by reintroducing this microRNA 15A. The reason we call it mimic is because we modify this microRNA by replacing the uracil with the 5-FU, which I alluded early as one of the chemotherapeutic drugs for treating solid tumors. And by incorporating 5-FU into this tumor suppressor, basically, you could imagine we basically build a stronger break, the break that's much stronger than the original. So that would provide the stopping power to eliminate these cancer cells. The reason it can do this because it can target multiple oncogenic targets, not just one of them. And a unique feature I'd like to mention to you is this microRNA mimic can be delivered to tumor cells without any delivery vehicle, which as scientists in this field, they would appreciate. This is a major bottleneck to deliver RNA-based drugs to tumor cells because it has to be delivered with a delivery vehicle. We all get COVID vaccine, which is an mRNA vaccine. It is an RNA-based drug, and it has to be packaged with a delivery vehicle shot to the arms to be able to function. Otherwise, the mRNA will be degraded. Similar things, the bottlenecks is here. And by modifying microRNA with 5-FU, we significantly reduce that burden. And this is an example how the microRNA mimic 15A is so effective in eliminating colon cancer metastases using an animal model. And you could appreciate from the image, and the cancer cells are labeled in red, which fluorescent under the imaging device. And the left side panel is control, which is not treated. And the right side panel is the mice treated with this miR-15A mimic. As you can see 2 weeks later, and we do not see any metastases with the treated groups, suggesting that these microRNA 15A mimic is extremely effective eliminating these colon cancer cells in the lung metastases. And the slide's bottom shows how this microRNA 15A mimic works. As I mentioned, it's a multi-targeted entity. It blocks several important targets such as BCL2, BMI1, and by doing so, it blocks many phenotypic changes such as invasion, metastases, proliferation, and cell cycle, as well as cancer cell stemness. Another example that we did is in pancreatic cancer. As we all know, pancreatic cancer is one of the lethal cancer among all cancers. The survival rate, 5-year survival rate is less than 10%. And so we use the same approach. We established the lung cancer metastasis model using a red fluorescent labeled pancreatic cancer cells. And as you can see, the left side is a control group, which you see obvious tumor growth. The next group is the mice that treated with a classical drug called gemcitabine, which is a chemotherapeutic compound that's being used currently to treat patients. And the third group is the mice treated with our newly developed miR-15A mimic. You could see that group, the metastases of these pancreatic cancers are significantly reduced. And the last group is the mice treated with gemcitabine, which is a chemotherapeutic drug, plus our microRNA mimic. And you could see nearly all metastases were blocked. And this really demonstrates the proof of concept of how powerful this microRNA 15A-based mimic has a therapeutic potential. And one thing I also want to highlight is the group of mice treated with this 15A mimic has no cytotoxic effect compared to chemotherapy. As we all know, chemotherapy has side effects. And we have not observed that in these mice treated with this 15A mimic. Thank you for your attention. Please submit your questions to coe@stonybrookmedicine.edu, and I'll be happy to answer them. Thank you.